Welcome to the 1 o'clock conversation today in Grand Ballroom 3. I'm Alan Sakian, one of the MCs for the evening. So we are going to be talking about terpenes and the entourage effect more than the aroma. Terpenes are a lot. They have a lot of different nuance that's going to be unpacked. You have a really epic moderator who is going to introduce your panelists as well. Michael Clemens, president and co-founder of Guild Extracts. Without further ado, let's pass it over to Michael.
Thank you very much, and thanks all of you for showing up today. We do have a lot to talk about with regards to terpenes. First, I definitely want to thank New West for bringing us all together, and definitely thank these panelists for coming together and having this great conversation. Before we get started, I'd kind of like to dedicate this discussion to Mr. Vanoni. And Mr. Vanoni was a 9th grade high school science teacher of mine, and he fueled my enthusiasm about science and introduced me to the scientific method, which was, at the time, it's an inductive approach to forming a belief system. And, it's kind of a play-by-play on how to vanquish intellectual opacity. I do want to recall a little story. I'll never forget in the spring of 1986, and I was excited to go up to his desk after class and show him some unexpected yet reproducible results from an experiment. And as I enthusiastically opened my laboratory notebook, I had a doobie half smoked to do a triple gainer with a twist and stick the landing right in the middle of his desk. And he and I both looked at each other pretty mortified. And I thought, I'm going to juvie, And he picked it up in his hand, and he looked at me with a look as serious as the heart attack that ultimately killed him, and told me, I know how much you love science, and you were born a scientist, but this is going to get you nowhere. And so today you need to make a choice. And so I'm Mike Clemens, president and co-founder of Guild Extracts. And here today with this panel of people to talk about the science of terpenes more than the aroma, which is probably better than my suggested title of, hey, you smell this? Or what's that smell? So I'm going to let my panel introduce themselves and tell you a little bit about what they do and contribute to this industry.
Hi everybody, my name's Samantha Miller. I'm the president and chief scientist of Pure Analytics. We're an analytical laboratory here in California doing Title 16 testing and quality control testing for the entire cannabis industry and anything you can put cannabis into. We've been around for about 8.5 years, and our focus is on providing the highest integrity of results, the highest quality, and an extraordinary level of accuracy.
Hi, I'm Jeff Raber. I'm the CEO and founder of The Workshop. We started as an analytical testing lab, and today we provide consultative services to license holders along with IP and technology licenses. We work with manufacturing, brands, and testing laboratories to help them get their scientific backends well-formed and well-founded.
I'm John Cooper. I'm the founder and CEO of a company that's called EBU. We're one of the leading cannabinoid research companies globally, specializing primarily on the pharma side as well as on the genetic side of things. As it relates to this, one of the things that we've been working on for a long period of time is actually proving what specifically is the entourage effect.
There we go. My name is Jeremy Plum. I'm the Director of Production Science at Proof Cultivar and a partner in Groundworks Industries. I'm a founder of a dispensary called Pharma in Portland, Oregon, co-founder of the Open Cannabis Project, and co-founder of the Cultivation Classic, which I'll speak about briefly, Oregon's largest craft cannabis event.
Great, thank you very much. Well, terrific, guys. I guess let's get started with, well, what exactly are terpenes, and why should we care about them? For anyone who doesn't know, who's been living under a rock.
I'll start out with in the most simplest terms, and we can progress in complexity maybe as we go down the road. Maybe that's a good way to start. So in the simplest terms, terpenes are the flavor and smell compounds in the cannabis plant and in a lot of other flowering plants as well. People have become really interested in terpenes because there's evidence and analysis that suggests that terpenes have a significant contribution to the overall experience that you have with cannabis in addition to the cannabinoids like THC and CBD and CBN that are present. So as people get more and more interested in the experiences around cannabis, in addition to the therapeutic benefits, which it's also seen that terpenes have significant therapeutic benefits as well, people are really interested in understanding more about the specific activities of individual terpenes, as well as the combined effects of groups of terpenes, the total amount of terpenes, and what that impact has on, say, the permeability of your biochemistry for the effects of cannabinoids.
That was a good start.
Next question.
No, we'll get a little bit more complex and say that the word terpene covers about 20,000 molecules that we know as organic chemists. And in cannabis, there's somewhere between 150 and 200 different ones, and they're comprised of repeating isoprene units. So little building blocks that get built up, bigger and bigger molecules, and then they get functionalized into terpenoids, which include alcohol types of functional groups. And they do impart the smell and physiological effects that we are getting, and plants use them to communicate with their environment and fortunately interact with us and or ward us off, such as some sort of irritants in some ways. So plants are defenseless little guys that have to stand there throughout their environment, and they need to communicate with the world in some fashion, and terpenes are typically what they use to do so.
Do we have any cultivators in the audience? Anyone here? Cultivation? Yeah? You ever wish your plants could talk to you? Yeah, they do.
They do. We just have to translate through the language of chemistry.
And I guess for the second part of the question, like, why do we care about them? I mean, we look at kind of like THC as an example. It wasn't really until studies in 1963 that we were able to put our finger on what exactly in cannabis was presenting the magic for everybody. It seems like the discussion around terpenes, at least in the consumer space and in the space in general, was kind of late to the scene. Why are people so infatuated with terpenes now?
Well, I think as Samantha talked about earlier, we're finding that terpenes have a very significant role to play in the experience that a person has, either for recreational type of experiences or for medicinal benefits. And if we really think about it, a person's not going into the dispensary today, walking up to a bud tender and saying, hey, I want to get high. Right? I mean, they're walking to a bud tender and they're like, look, I'm just looking for something to help me chill out, right? Or, hey, I'm going to go skiing, or, hey, I got to go vacuum my house, right? I mean, they have all of these different reasons, right? And so part of one of the major things that we're trying to understand is if we're going to make a mainstream cannabis culture, right? What's going to be very important is how is it that we can deliver consistent, known, understandable effects for those end consumers. Because if the person's coming in, and wants to go out and party with their friends, but they take something and it makes them want to pass out, that's just not a very good experience, right? And so the more that we can understand about the compounds that actually cause the specific effects that people are looking for, and further understand how is it we can actually deliver those compounds in a consistent manner so that we can reproduce those results again and again and again, the more likely we're going to be able to create a mainstream cannabis marketplace, and the more likely we're going to be able to actually create real medicine that's going to change the lives of hundreds of millions.
Yeah, so it's like I've been asked before, do people like shop for cannabis, by terpenes? And it's kind of funny because you go back to the day, let me flash back to 1986, that doobie that did the triple gainer with the half twist. I probably shopped by terpenes. It was the first thing you do. You get a, cannabis as you open it up and you smell it. And so I think that just kind of intuitively we can kind of learn a lot about the things we put on our body by our sense of smell. And this is something that we've kind of cultivated over thousands, if not millions of years of evolution.
I think one of the interesting experiences was at Pharma. We were the first dispensary to actually have full terpene profiles actually at the shelf. And this notion of like reorienting consumers towards more targeted and reproducible effects was kind of the baseline of our operation. We would test with only one lab with all of the flower we received. We had real apples to apples results in a time where lab to lab consistency really wasn't an option in Oregon, and tried to have as much detailed information about terpene profiles as possible. We saw an evolution in the kind of consumer we were serving in a very short period of time. We served more women than men, more people 50 and up than under, and rose to become one of the most successful dispensaries in the state. But I'd love to share one of the practical examples about why are we continuing to go down this path from the Cultivation Classic. This event has gone on for 4 years, takes a lot of lab data, much like Samantha's work with the Golden Tarp Awards. We have a chemist in Oregon who's essentially taken all of the samples from producers everywhere. We also have a partner, Dr. Adi Poe, who's a neuroscientist. And Dr. Adi Poe has a company called Habu Health that's been doing investigations about qualitative patient data. And so we have this judging process, which is not a normal cannabis cup evaluation, where essentially the judges are trained to input feedback about their experiences. But we have this really strong chemotype baseline data, and we saw two things this year after we took all of the data from the dashboard and did causal analytics with a machine learning team that we're partnered with. The two things that we took away were completely corroborated in Ethan Russo's work and a variety of other people's work based on what we've been reading. It was fascinating to reproduce it with our own process. With more than 160 responses and samples correlated, we saw essentially 3 compounds associated overwhelmingly with positive mood inducement. And that was pinene, limonene, and beta-caryophyllene. Whereas we also saw this other result around cognitive performance enhancement associated with terpinolene. This is, of course, a small data set, but it's very, very high quality. We are used to seeing a lot of dirty data at scale and still kind of reaching, but it was just neat to start to see that we could start to associate effects in a broad way and offer more targeted, consistent cultivars, hopefully, and chemovars for a discerning consumer.
Yeah, you mentioned Mr. Russo's work, and in his 2011 study, he did cite an article, the Buchbauer 1993, Fragrance Compounds and Essential Oils with Sedative Effects, where specifically it outlined where mice that were exposed to terpenoid odors that were inhaled from ambient air for 1 hour demonstrated profound effects on their activity levels. For instance, linalool presented a 73% reduction in their physical motility. So it's like, couch lock anyone? And so now as you start adding these compounds together, you know, then we start talking about, well, the other kind of part of this discussion, which is the entourage effect. You know, and it's like, I know a lot of people have heard about this, but it's kind of elusive on exactly what is the entourage effect, how does this work, how do these compounds act synergistically with each other to kind of change the overall effect or present the type of experiences that we get varied from phenotypical plant to phenotypical sample. Samantha?
I think that's the key cannabis question, right? So, you know, when you see linalool causes that effect, is that's an extrapolation of just one molecule, and is that exactly what happens when you have that molecule amongst many others in a cannabis composition? So what we see is complex interactions of many molecules, and there might be something else present in the plant that increases motility. So if you might have some linalool, but you might have something else, maybe that cancels out and or amplifies and or, you know, does something differently than it does individually by itself. So a lot of the studies that we have up to this point are limonene does this by itself, linalool does that by itself, but we don't know what they all do together, and we certainly don't know what they all do together when they're with cannabinoids. So understanding the composition and then leading that towards which effects we're going to get is kind of the next evolution of what we're really all up to today.
Now, when you're looking at these, you know, these samples with these compounds in plurality, I mean, I guess you also have to consider that the human body itself is an entourage effect, isn't it? And so when you start changing, you know, you have, you know, people all have their own unique biochemistry, and they have their own compounds in plurality. And so I guess it would probably present quite a challenge for people really trying to, you know, put a tack in what does what, you know, when it's such a complex matrix.
Yeah, if I could comment on that, I think it's one of the most interesting things in my own work with patients. So in formulating various strain-based terpene profiles in varying amounts of THC and CBD, I put various formulations out into about 750 patients and got self-reported patient feedback on what their effects were around sleep, mood elevation, anxiety, sexual arousal, et cetera, et cetera. What was really interesting was, you know, when you look at the animal model research and we see things like linalool is correlated with this, limonene is correlated with this, terpinolene, et cetera, we take those as kind of a starting point, as maybe a suggestion of what might happen when those compounds are present. But what I saw really clearly in the work was you certainly can have what I call drivers, right? So when linalool is in a terpene formulation, it's absolutely a driver towards sedation, especially when it's paired with alpha-terpineol, especially when it's then paired also with myrcene. And so what you start to see also is the additive effects of terpenes that have similar effects like alpha-terpineol, linalool, and myrcene on the sedation side. But what was really interesting was sometimes there would be, say, other terpenes present, especially in the sesquiterpene side, like farnesene, which also can be quite an experience driver along with neurolidol. When those are present in higher amounts, sometimes you see actually different effects than you would expect based on your your observations when those other terpenes aren't present and just the ones that I mentioned before were. So it's definitely the overall combination of terpenes. It matters what the relative amounts are. There are definitely drivers, but those drivers can be almost quenched by other terpenes with stronger influences. So it's quite
I've heard
dynamic.
that like the effects that these things can have are can be plotted on a bell curve. So you're on one side of the bell curve, it's going to have one effect. The other side of the bell curve, another effect.
Absolutely, and I think you see the dose-based effects with both terpenes and cannabinoids with a lot of patients.
And John, you guys at EBU have kind of, really set your focus toward creating this layer of predictability between products in the marketplace and the customer. And so how does, how do you guys put your thumb on this very complex issue?
Well, we realized about 4 years ago that if we were just doing human studies, it's a little bit like shooting in the dark. There was just so much going on in order to do that. So we ended up creating a drug discovery lab, which we're actually doing high throughput screening. So the easy way to think about it is we actually grow live human receptors in-house. So CB1, CB2, serotonin receptors, capsaicin receptors, we're getting into opioid receptors, dopamine receptors, et cetera. And then we take precise combinations of purified cannabinoids and terpenes, dose those on those receptor sites, and can read in real time and measure the biological reaction of what happens at these receptor sites when we do this.
So you have a brain on your desk in the laboratory, is what you're saying.
Yeah, I learned early hire brains. It's very good. I'm the only non-scientist up here. The only thing I'm good at is hiring scientists. This is what I do, right? So the reason why this was so important for us was because the measurement of what's actually happening at that cell site, right at the receptor site, where the cannabinoids and terpenes are actually interacting with that cell, really matter. And the complexity around this, I mean, most people have this perception of biology that you learned in 10th grade, where the teacher put a cell on the board and they said, here's a receptor, and something came and plugged into this receptor, and this is actually how we think of pharmacology, right? And this is reinforced by the world around us. So if you have a headache, you take ibuprofen, right? One compound's designed to solve this problem. Well, the world of cannabis, you have to think about it very different, and primarily because most of the targets that we're looking at in the body are what are called G protein coupled receptors. And the way to think about those is that they collect lots of different compounds at the same time, and depending on that combination of compounds, you'll see a very different effect. This comes to life because like your taste receptors are G protein coupled receptors, right? So depending on the combination of things in your mouth, right, you can actually taste lots of different things, right? Chocolate tastes like chocolate, peanut butter tastes like peanut butter. You put it together, you actually get a different sensation. And so we started building this from the ground up, right? You create the foundational reaction of what happens with one compound, but then you actually start looking, what happens if we use these two compounds together? What happens if we use these three compounds together? And a quick example of how powerful this is, most people think, if you have pure THC, that's going to be the highest high that you have. Interestingly, if we take pure THC, when you apply it to the CB1 receptors, the one in your brain that actually makes you feel high, we only see about a 40% reaction on that CB1 receptor if we were normalizing the way we score data. So it's what's called a partial agonist. If we instead of that, reduce the amount of THC, bring it down to 70 to 80%, and we add in a couple very specific cannabinoids that we've discovered and a couple very specific terpenes, we'll boost the reaction on the CB1 receptor to over 80% while tripling the potency. If we choose a couple other very specific cannabinoids and specific terpenes, we can bring down that reaction to under 15% and wipe out the potency, right? So what really matters is not that you have a bunch of terpenes, right? What really matters is you need to know the exact terpenes, right, and exact cannabinoids in order to get the desirable effect that you want. And what's interesting, the discussion we were talking about earlier in terms of the body being a very complex thing, we've now actually run over 50 human studies. And what was interesting in this human studies was if we create a formulation from scratch using these purified compounds, and we're trying to go after an effect like chill, it didn't really matter in our studies if a person was black or white or male or female or young or old or heavy user, light user, whatever. We could almost get everyone in double-blind studies to report that they're feeling chill. Right? We could make people feel create. We could make people do all these things. So, the reactions that people are feeling are much greater than the placebo effect, right? And the complexity, we're going to see a lot of complexity if you're trying to use every compound that's in the plant, right? We just call that chemical chaos. But if you get down to just the right specific set of compounds that are going to create the right desirable effect or the right specific medicinal reaction, right, you're going to be able to get the vast majority of people that actually sit in that bell curve to have the desirable effect that you're looking for.
Great. Well, thanks. That's quite interesting, and that kind of feeds into the second, kind of the second side of this, which is like, you hear a lot of talk out there in the space about cannabis-derived terpenes versus terpenes that are sourced from other plants or other sources. Is there a difference between these things? I mean, is
Molecule's a molecule. So
I'd
the
have
high
to
purity
agree.
molecule is molecule.
Regardless of what you hear, CBD from one source, but of course it's like, you can say, well, this guy is always this guy, but sometime, and every time he's at my party, he does great, but when he brings his friends,
Right.
And so a molecule is a molecule, but his entourage, however,
Right.
may
It's
vary
what else
from
goes with
one
that,
side to
right?
another.
So in cannabis-derived terpenes, do we know all of the fates of those original terpenes? Have they isomerized, oxidized, degraded into some other unknown Franken-mixture type stuff? Even though they came from the plant, it doesn't always mean it's something healthy or safe for you because we didn't know what they became. If you understand each individual component and you can reproducibly put them in there, you can make that look far closer to the actual plant composition than you would by just extracting them. So I think a lot of the perception is cannabis-derived terpenes must be safer for me because they're from the plant. While they're from the plant, we also don't know everything else that they became, and we don't know all the toxic profiles of any of those things either.
Toxicity is always an amount, isn't
Right.
it?
It's a dose response. What else is with it? Maybe something there is to protect against that as well. So understanding the chemical compositions of those mixtures and how we got there and can we reproducibly get there is really what would be important for standardized medicine.
And really, I
understanding
can
the
see
approach.
that, trying to understand a particular entourage is quite challenging because it's my understanding that the amount of terpenes that we can actually test for in the most sophisticated testing laboratories is still only a fraction of what we know to be present in the plant. Do you want to speak to that a little bit?
Well, sure. I mean, as Jeff commented earlier, with 150 to 200 different terpenes that are supposed to be in the cannabis plant, right now we have a list that spans 45 compounds, which is considered an extensive list from an analytical laboratory, but still, again, is just a fraction of what's in there. There's actually a scientist in Israel, Dr. Dedi Meri, who runs what's considered to be the world's most advanced cannabis laboratory at the Cancer Biology Institute at Technion University. And Dedi has just an incredible luxury of a massive budget, a lot of PhDs, and like 30 triple quad mass specs. So we're all quite jealous of Dedi and his laboratory. It's quite beautiful. And so he has the ability to do analysis that can look for all of these compounds, whereas in a commercial laboratory like mine, which is a service-based laboratory, we also have to do things that are profitable, for example. And so running an analysis for 200 compounds is a multiple-hour analysis, for example, where you can only do a few of those a day. And so in the commercial realm, until there's a lot of interest and a lot of value placed on that, you probably won't see services available.
So you stay pretty late, don't you?
We do stay late.
And has lots of machines.
I do want to say, though, there is, based on everything we've seen, there is still so much opportunity, both on the medicinal and the recreational side, with even the top 20 terpenes that come from this plant.
Oh,
right?
for sure.
So,
And I think that cannabis science in general is so new. I mean, we've had this, these regulatory intricacies for the better part of a century. And, we have the NIDA, which is kind of in control of all the research cannabis. And then, of course, the Drug Enforcement Agency, which is the teeth of federal drug policy. And you have to kind of petition both of them if you want to engage in a study on cannabis, and they'll send you back a form letter that basically says it's not in our mission to look for utility in a Schedule I controlled substance because there is none. And so it kind of presents challenges from a scientific side where it's hard to put things down an FDA product pathway like, most things, and so we've had to really, as an industry, kind of fall back on crowdsourcing wisdom in order to, kind of better understand what these very complex matrices are doing to people. And Jeremy, you've talked a lot about this, and I've seen your talks before. You talk about wayfinding in this space and kind of a little different approach to crowdsourcing this wisdom. So you want to share with us, because your approach with Proof Cultivar is, you're kind of giving nature a helping hand on creating these profiles that also seem to have fairly predictable, if you have strong control over your genetic collateral, you can get fairly predictable results, thus also offering a layer of predictability between your flowers and the customer. Do you want to talk about that a little bit?
Certainly. I do want to pick up very briefly the distinction. We just saw this article come out about LaCroix featuring non-natural additives. And of course, if anybody read this piece, we talked about linalool as a cockroach killer. And there was, to the horror of all the people who had never heard of this scary compound before. But I think the real distinction with the synthetically derived terpenes at this point has to do with product quality. It's that we find that, if we're taking things that are the outcome of petrol distillate and we're having kind of 5% other compounds that are included, and then they're stacking as we include more and more. The plant-derived are very favorable just from product purity perspective, and I just want to make sure the distinction between the synthetic sourced and the naturally derived products.
You can get natural terpenes of like 50, 60, 70% purity too, though, and there are other things that could be with them that are not what you want nor what you would prefer to have. So the word natural is a very, very dangerous word, and that's where all the lawsuits are coming from. I choose the third definition in the dictionary, found in nature. So it doesn't mean that where it's derived or where I'm getting it, but it is found in nature, and then we can always replicate that in many ways.
Key point. Oh.
Yeah, I was going to say, well, I think one of my own observations is really the distinction, not so much towards industrial versus cannabis derived, but synthetic versus botanically derived. Because in the case of synthetic terpenes where they're actually being synthesized and there's catalysts present and other chemicals that are used in the actual manufacturing, nothing is ever manufactured to 100% purity when you're talking about organic chemistry. So there's always a percentage, whether it's 1% or 2% or 3% or more, that's other substances. In the case of something that's synthetically made, that 1 to 5% are other chemicals. That may not be something that's very palatable, for example. In many cases, that's what you find with synthetic terpenes when you try to include them in vapor cartridges. They often have kind of a sharp taste to them, and you can tell that there's something chemical flavor in there, right? So in the case of botanically derived terpenes, whether or not they're industrial or cannabis derived, tend to have a lot more palatability and less of that sharp chemical flavor because that 2 to 3% of non-pure terpene that's in there is either maybe other minor terpenes or other co-extractables from the plant versus chemical substances and catalysts.
I could take a botanically derived mixture, though, and melt your face off with it. Like, there's no way that that's not an indicator of purity. So some of those compounds are very sharp, they're very harsh to the tongue, and depending upon their concentration in your mixture, whether they were naturally derived, synthetically derived, still going to taste terrible. So it's not always an indicator
for the
of
average
purity.
user who goes into a dispensary, they really don't have any way of determining, like, does this have cannabis-derived terpenes? Does this have, industrially sourced terpenes? And when you look in the marketplace, I mean, if you go to Amazon.com and you buy a 1-ounce bottle of terpenes for $8, or you can go to another website and get cannabis-derived terpenes, $200 for 1 milliliter, which puts it right between like the third and the fourth most expensive perfumes in the world.
Which, following up, I love being on a panel.
I mean,
with
is there a
two of
value
the
there, or is this hype?
leading analytical chemists in the state, and ultimately having that level of elaboration. This is a joy. One thing I would love to point out is that right now we have an industry that's, at least in the United States, certainly related to academic disciplined research with pharmacokinetic and dynamic studies and disciplined human trial work and so forth. But at this point, what I see predominantly is actually a lot of companies going forward that are, product-producing companies that are doing in-house kind of walled garden science that aren't really sharing the secret sauce with other companies and then going to market with targeted effects. When I wonder if we have come to a point of maturity in the research where we're able to talk about targeted effects to the extent we do. And when I look at the incentive to market and sell products and to kind of come to market and arguably race to market with that first mover advantage, I just am concerned about the implications for human health and well-being when I think of another chemist in Israel, Lumir, who visiting Lumir is a thing of wonder. He's been going since the 1950s doing cannabis chemistry analytics, and he's telling me he's characterized more than 700 compounds. And that really, when we're talking about aroma profiles, from a producer perspective, you're trying to describe the fuel profile in an OG Kush plant. Good luck mixing and titering the right amount of myrcene and limonene to produce the fuel nose. And in fact, in order to get to a deeper level of insight, we of course have to deal with all the substrata of chemistry, including aldehydes, esters, ketones, flavonoids, and you need these variety of other compounds to actually create profiles like cherry, berries, banana. A lot of the terpene profiles that people are drawn to include a lot of other chemistry. And the truth is, I believe we're at a point where I think a lot of long-term research is needed to substantiate that we can extract and refine a few compounds and disproportionately favor them when we don't understand, as Samantha was saying, all the interactions and the complexity between these different products. So what I've taken on as a personal mission after seeing the huge amount of product diversity and quality issues in the marketplace was trying to address the supply chain dynamics from the whole plant efficacious perspective, which is that you have to absolutely use precision agriculture in the greatest steps to be able to have reproducibility. And we're talking using, for example, at Proof Cultivar, we're looking at like 50,000 genotypes for every seed we crack in this new phenotype discovery program we're working on. A machine learning-based environmental control solution that has very, very high-resolution sensors that will culminate in a lot of three-dimensional sensing and comprehensive analytics of the environment. We have all the cultural practices, and then moving more into chemical fingerprinting or comprehensive analytics to really unite that chain of events to understand these kind of baselines that I feel that no producer has been able to really grok. And for example,
well, with that said, like how much have you determined, the whole nature versus nurture? Because like we have found, like we have a strong analytical core at Guild Extracts. We do our own laboratory testing, in-house to try to get as much information about the product before we get started. There seems to often be a wide variance in the constituents that are in the bottom part of the plant versus the top part of the plant. We've actually seen certain terpenes that only show up when a plant has been exposed to starlight, which is interesting, but at the same time it isn't, because like plants don't have eyes. Why would you think the electromagnetic spectrum stops at visible light with them? And so in your findings, what have you found, like how much of the terpene expression or this phenotypical expression is really nature and how much of it is nurture?
I really appreciate that you brought this up. As a producer with analytical testing access for almost a decade, I'm a very lucky character. I think it's very recently that we have the resources that Samantha and Jeffrey and others are providing to provide this insight. And what I saw first was if I gave an exact genetic copy, a cutting that I had closely monitored and gave to 4 different producers, and then we did really intensive analytical work on that, we saw an incredible range. The reality is in one sample, there was 28 milligrams per gram of beta-myrcene and another 2, and those 2 products are going to vary widely with the exact same genotype. And there was a huge amount of distinction in the production environment. I've since reproduced that work at scale at Proof, and one thing I can tell you is that the epigenetic or the phenotypic expression of cannabis and the excellence of both the technology, the facility, and the plant itself are going to cause the ability to take one single cultivar and create a range of products varying only the light diet and the other environmental inputs. I just came yesterday from a conference in Chicago called PhotoX. It's the leading photobiology conference. I will say, from a production perspective, the wavelength is a massive variable in the production environment, and we're at the absolute edge of finally, with the advent of LED technology, being able to isolate specific wavelengths, explore at different stages of development the impact of those wavelengths, and connected with photon intensity, once you've provided for all the other photosynthetic needs and the baseline needs for the plant health, this is going to be the single greatest variable in expressing phytochemical diversity from my perspective at this point.
Gotcha. And I think one of the other challenges that comes up is like, if I had $1 for every time someone came into my analytical core and was like, she handed me some cannabis and was like, what's in this? And it's like, it's not really how testing laboratories work. You don't have a magic machine that you just put a bud in and it tells you every compound that's in there. I guarantee you would like to have that machine, wouldn't you?
It'd be a lot less expensive.
Indeed, indeed. You have to really know what you're looking for. And in something that has such a complex matrix, I mean, that must present a tremendous challenge when it comes to kind of looking for, again, to try to create that layer of predictability between our cannabis and our, experience.
If I may say one more thing, and I think this is maybe the most important thing, is that when I was at ICRS this year, I saw one person capable of stepping forward in the International Cannabinoid Research Society from a whole plant perspective, saying, I can systematically produce this chemovar, which I have bred to be a type 3 plant that's a 1-to-1 of beta-caryophyllene and CBD. And this is a really amazing therapeutic outcome, and that then can be relevant in the much bigger conversation in the pharmacokinetic and dynamic studies and drug development programs with all of the pharmaceutical companies that are represented there, and that literally everybody else was talking about synthetic cannabinoids at this juncture, and that there was this huge amount of people there. And I think one thing I know as a producer is that we have this very limited period of time where I think we can mature a lot using all of this advanced technology, and there's really an important place for controlled environment, precision agriculture to simply still be at the table in what is the biggest market share in this whole space. What is the most important high impact and long-term expression of cannabis is to actually continue to derive meaningful but consistent expressions of a whole plant therapeutic efficacy, and to have that continue to be a part of the conversation for many reasons, from an ecological perspective, but mostly from a humanist perspective, that people and plants have evolved together for a very long time, and that I want to see a future where the plants continue to play a really meaningful role in this most lucrative sector, but that means a lot more emphasis on
For
this
sure,
sort
and I
of-
think you're touching on something important, is that, you know, we're doing a great job of kind of covering a lot of the challenges that we face from within the industry, but, you know, this presents a whole new layer of complexity for folks who are trying to get that themselves. I mean, as soon as you walk into a dispensary, you know, you're going to hear the same thing from pretty much any bud tender: indica, sativa, or hybrid, which is basically saying, do you want a plant that's been agriculturalized by man? Do you want a plant that comes from south of the 30-degree north latitude on the India subcontinent, or, you know, a mix between the two? I mean, are there any indica strains, sativa strains? Is there any correlation between a plant's morphology and its effects?
Absolutely not. And I can
You know,
show you
and-
a range of variables with broadleaf cultivars that have what we would consider to be exciting chemistry, and then narrowleaf cultivars that are utterly sedating. And the bottom line is all of the distinctions in the morphology taxonomy debate, I believe, have been eclipsed. The old definitions should be updated. I think the work of Rob Clark and George Merlin and cannabis ethnobotany and evolution is the most meaningful that I've personally related with, which deals with narrowleaf drug cultivars, narrowleaf hemp cultivars, broadleaf hemp cultivars, narrowleaf hemp cultivars. Bottom line is there's these 4 categories and then putative ancestors. That deals with plant morphology, but the problem is that's really botany speak for plant geeks. And that's useful. What's really useful from the consumer and patient perspective is the phytochemistry and the chemovar perspective, which we're talking about.
Well, how do we, like, as professionals, bridge that gap for the consumer? Like, for instance, you know, if I go into a dispensary, you know, it seems like half the time they've made up a strain name because they have the same strain on the board. And so it's like there's a certain sense of responsibility on our side as a producer to, like, do that work for people. And Jeff, I know you, you know, some time ago kind of made the shift at the workshop from being just an analytical testing laboratory to actually, you know, formulating products. And, you know, maybe you can speak to, like, how much have terpenes and stuff changed your workflow in like kind of, you know, creating these products for customers, or again, kind of trying to create that layer of predictability between, you know, the product and the consumer.
Dramatically.
Yeah.
Yeah, dramatically. So they're a major focus of driving effects, driving the entourage or ensemble effects, and really creating
I
consistent.
love that ensemble effect.
Yeah.
I like that
Well,
a lot better. It's Dr.
better than
Lester
Entourage,
Grinspoon.
right? Yeah.
And, you know, Entourage to me always sounded like THC is the star and everyone just follows
Just
him
the one
around.
guy.
And again, I think it's a
all
lot
of
more
them.
chill the other way. It's like, no, we're just hanging out together, doing good things.
I like
Thank you
ensemble
for
better
that.
too. Yeah. I mean, it's really the major focus of what we do to drive differentiating effects. You have a few cannabinoids that you can play with consistently. There are more rare ones that are starting to emerge, but how you are driving most of what you're seeing in terms of differentiation comes via effects, comes from terpenes. So understanding them, putting them together in the most complex and best standardized fashions are really what are driving, I think, next generation type products. And it's really been learn from the analytical lab, learn from what the plant teaches you, and then how do you utilize that to create the best standardized products that will drive medicinal or adult type product effects. And I think truth in labeling is what we're kind of all kind of hinting about and talking about, like, where did my product compositions come from? Where did my ingredients come from? How do I designate if it's, you know, not indica sativa? What words are we going to use to really drive education for consumers and proper communication with them, and then doctors and others amongst the community? Because only if we use the right words and right terms that everyone understands in a good sense will we actually be able to advance our cannabis conversation.
I think there's an important paradigm for everyone to think about, though, and something I've kind of been challenging people to think about as it relates to your own company, which is, are you a cannabis company or are you a cannabinoid company, right? It's a very big difference, right? Because, you know, there's definitely one path where you're trying to create the exact same set of compounds that are going to be in this flower every single time and have a pretty good idea of what it is. but you're still dealing with nature, right? This thing is still chemical chaos. There's going to be a high level of variability, and at least from our perspective, that's going to be very hard to control. But you also have lots of different compounds that are doing competing things when they go in the body. I mean, you're dealing with dozens and dozens of different compounds that we actually call dirty compounds because they're interacting with lots of different receptors in your body at the same time. Versus the cannabinoid company is the company that's trying to create something from the ground up that's just going to be a standardized product using just a specific set of compounds that are going to be 100% reproducible every single time, right? And in that world, that world in terms of how is it we're going to be cultivating plants and these compounds is going to look very different, right? We're not going to spend a bunch of money, trying to fix the lights and do a whole bunch of things in order to get this exact same plant. You know what we're doing? We're going to be setting up tens of thousands of acres of primarily hemp designed to basically grow a specific compound at a fraction of the price of what everyone else is doing, right? Same thing on the terpenes, same thing on everything else, right? And that's where I think you're going to see this kind of divergent path that's going to be happening in the world, where you're going to have cannabinoid companies who are very much going to be building these very precise, exact products every single time, where the inputs are coming from a commodities market, and then you're going to have what I call cannabis companies that are going to be really looking at the full spectrum that's actually coming from the plant. I'm not arguing one way is necessarily better than the other. I actually think both of them will be in the marketplace for a long period of time. But I think it's very important to understand there's definitely two different paths that are basically emerging right now in the marketplace, and they're going to continue to go down two very different paths over the next couple of years and completely change the economics in this industry.
Do you think that these
That
paths are
sounds
truly
like indica
divergent,
sativa
or
talk.
because, like, have we had anybody walk into a dispensary and say, "I'd like some cannabinoids"? No, everybody goes in there looking for cannabis, and so a lot of this is kind of happening behind the scenes. And I think that people will go into dispensaries and go, "Look, I'm looking for a product that's going to have consistent results. I want to know, like, if I go and I utilize this product before going to a concert, I'm not going to fall asleep halfway through." And so having that layer of predictability is important, but we still kind of have one kind of consumer. And
yeah, but I think it's going to probably even show up more in medicine. Right? I mean, for anyone who's kind of in that 55 plus crowd, when you do the interviews on them, most of them aren't looking to get high, right? They're just looking to improve their life.
100%.
Right? And if we can deliver something that's going to be 100% consistent, that they're going to get the relief they need, they're not going in there looking for, green crack or AK-47, right? They're looking for anxiety relief. They're looking for pain relief. They're looking for these type of things. And if we can, it doesn't matter the store they walk in, right? If they could go in and get what they need every single time and they can rely on that changes their lives.
And I think that this has been a philosophy within, from producers for a long time. In fact, Samantha, you worked with Humboldt for a while. And I don't know what your role was there during the time, but back in 2016, they made the dose pen, which actually gained one of the best inventions of 2016 for Time magazine. And so this discussion isn't a new one. This has been going on for a little while. Do you have anything you want to share about your time
Sure.
there? And like, were people talking about terpenes back then, or if you mention terpenes, they look at you like a cat looks at a new gate?
Thanks for the question. So I was the founding chief science officer at Humboldt, now called Dosist, and my role there was to design their formulations that were supposed to elicit a specific effect, whether it be euphoria, sleep, sexual arousal, anti-anxiety, et cetera, et cetera. So we took a slightly different approach to the products that were on the market at the time by doing specifically formulated cannabinoids and terpenes. So we were using botanically derived terpenes as individual molecules and reformulating full strain profiles and including those in the formulations. It was a time where people were definitely talking about terpenes, but less about the individual molecules and the individual substances. I think it was very much a 30,000-foot level then. And so one of the things we did on our packaging was actually list the terpenes and their relative amounts that were in the pen. And that was something that was very new at the time. I see it everywhere these days, but it definitely started a conversation. And in our conversations that were public-facing, we talked a lot about the combination of the effects between cannabinoids and terpenes and included it in the conversation. And that was something that was definitely not a part of a serious conversation at the time, more of a more terps, more fire kind of a conversation, I think, was mostly what was happening, except for in kind of discrete areas where you have companies like EBU and companies like Humboldt who were specifically focused on the specific effects of those substances.
And it's like, we kind of talked a little bit earlier about some of the challenges the industry's faced with regards to having a really, this institutional bias against the science of cannabis. And like, are there these other fields of science that we can draw meaningful information from. Like, I mean, aromatherapy is kind of a thing. It's kind of been a thing for like thousands of years. And, you know, there's been extensive studies that, you know, have found the pathways on how olfactory connects directly to the oldest parts of our brain, which then stimulate our endocrine system, which produces hormones to help our bodies stay in balance and a sense of well-being. And now here you have the endocannabinoid system comes onto the scene, and you know we find that that modulates our endocrine system. And so, could this be a mechanism on how these things work together? Like, do you have you know, the aromatherapeutic aspect of cannabis that kind of, you know, stimulates that endocrine reaction, and then you have the cannabinoids that come in and conduct how this all works? Any thoughts about that?
Yeah, I mean,
I figured that out when I was smoking weed, just saying.
I think that's a very interesting point, but I'm going to take it a step further, right? You know, high probability, even in those circumstances, those terpenes are getting into the bloodstream. And so not just the reactions that we're having actually from just smelling them, but when they're actually in the bloodstream, is there a direct effect on these terpenes at different receptor sites, not only just the endocannabinoid receptor sites. And so very specifically, if we look at serotonin receptors, there's actually 3 very specific terpenes that could play a tremendous role in the future of how is it that we're going to be looking at formulations related to anxiety and depression.
And so the serotonin receptors are kind of your feel-good receptors, you know,
Yeah, I mean, it's...
among other things.
Yeah, they're very complex set, especially when you're looking at serotonin receptor 1A versus 2A. And kind of the...
Number 5 is my favorite, by the way. Any biochemists out there will know why.
Very, very excited. Yeah, we're going to
Down
totally
with
geek
5.
out with all that type of stuff. And the simple way of actually explaining the, I guess you could say the ultimate formulation, the ultimate solution that you would want related to anxiety and depression would be if you can create a formulation or compounds that will actually upregulate the reactions that you're going to see at serotonin receptor 1A, while having the complete opposite effect simultaneously at serotonin receptor 2A. And there's nothing in any of the pharmacology books out there that actually show those type of reactions. We're actually demonstrating right now in the laboratory that we're seeing those type of reactions, both with the cannabinoids and terpenes that are coming from this plant and actually making this work. So we're, you know, as an example, we're very excited about the future of anxiety, depression, PTSD, and those type of opportunities based on a very specific set of compounds that are going to be coming from this plant. And so the point being, you know, the terpenes can play a specific role. I actually think they may actually play a more specific role just based on the work that we're doing right now, by them actually getting into the bloodstream and directly having an impact on those specific receptor sites.
Well, the
For
nose
sure.
is directly connected to the brain, right? So you can have that aromatherapy response, but if you're using a topical or even an oral dose and getting it into the bloodstream via inhalation, they can have different interactions than just aromatherapy based.
You know, and like the study that was showing, I mean, maybe you can give everyone an idea of like how small is, what was the number that I said? It was 4.2 nanograms per mil. That's a very little bit of a chemical. And another thing I learned back in high school is very small changes in your biochemistry can have very profound impacts on your experience. You know, a few micrograms of this, that, or the other thing, and your whole world is a different place. You know, and so, yeah, so...
Fine line between medicine and poison, that's the dose, right?
For sure, 100%. And I think that, you know, that people for a long time have tried to draw this fine line between therapeutic, you know, or medical marijuana and recreational marijuana, when really it seems to me that it's much more of a spectrum. I mean, it's like, you know, I don't know if you want to kind of talk about, you know, the difference between medical marijuana and recreational. I mean, for recreation, I'll, you know, go and I'll ride my bike, I'll play music, I'll hang out with friends, and these are all things that I can augment my experience with cannabis. But I don't smoke pot for fun, you know.
It's all cannabis to me. I think that like medical and adult use are legal recreational, or like legal labels, regulatory labels, tax labels, not
For
really
sure.
like
But this has been a
human
discussion
cannabis
for a long
interaction
time.
labels.
I mean, a lot of people in other states are saying, you know, well, I'm okay with medical marijuana, but no, not the recreational stuff.
Or I won't use that medical stuff till it's legal, right? So I've heard that as well.
I think that points to some of the stigmas, right? Some of the fears,
100%.
some of the fears that people have. I think that's where that comes from. I did a panel recently at a Berkeley Ideas Festival, and the last couple of public talks I've done, the people who have come up to me afterwards are moms of teenagers who are using cannabis, and they want to know what I think about it. They want to know if I think that's okay. They want to know actually if they should actually procure it for their teenagers because they're worried about pesticides and the things that their kids might get on the street. And so I think that when it comes to talking about cannabis and safety and what people are really concerned about and how the different ways we think about cannabis in terms of medical versus personal use, I think we really start focusing on stigmas when you have adverse reactions to personal use. There's some seniors who are afraid they're going to fall down if they use cannabis, for example. So there's fears, some founded, some unfounded, but that's where I think those distinctions come from. It reminds me of the phone call I got from the state of Florida 7 years ago, asking me to help them understand THC and CBD because they were writing their CBD-only law. And I tried really hard to talk with them about the fact that there wasn't a reason really to exclude THC, but they weren't in a place where they could hear it yet. And I think that what you see happening right now is the creation of an environment of permissions to be okay with cannabis, and it's kind of the very first step. And in more progressive cultures like the ones we have here on the West Coast and on the East Coast, you see a more kind of early adoption of that. But it's a slow tide that's definitely happening. But I think
Very slow.
A very slow tide. But the more actual factual information that we can provide and scientific information that can actually come from government research entities, because those are the things that the people who are concerned are listening to. So,
Well, for sure, and I think kind of talking about, and anyone who's heard me speak before, I've said it before, that we kind of like, when you look historically at what institutional bias against science yields, you can go back to the Middle Ages. And it was, the institution at the time was a Catholic church, and science was taking this inductive approach to describing our experience that, distracted people from these proprietary spiritual themes, and we wound up in the Dark Ages. And so that's kind of where cannabis has been. I think that some of the first discussions around therapeutic utility were happening in the 70s. There was that think tank that was put on, John Sperling and Soros, to kind of look for, okay, how do we get regulators behind legislation reform without being branded communists? And that was the think tank, and they came up with medical utility and wrote the playbook on Got to Find the Poster Child and so on and so forth. And here it is, half a century later, and we're now kind of coming into this renaissance. It's super exciting.
If you don't mind, can we bring up a really important subject? Jeff kind of hinted at it, but I think it's something that especially the scientific community in this industry probably needs to pay more attention to, which is, we know THC is super safe, CBD is super safe. It's basically nearly impossible to kill yourself by overdosing with those compounds. There hasn't been a ton of studies related to terpenes, and what we're seeing right now in the marketplace is people are increasing the amount of terpenes that we're seeing going into these products today.
Without visibility as to what the impact of that's going to be.
Right. We're actually, it's one of the things that we're concerned about because there's potential toxic effects. I think, the terpene levels you see in the plant today seem to be safe from everything that we're seeing, but, if all of a sudden 20% of your product all of a sudden is terpenes or 50% of your product's terpenes.
It absolutely have very negative consequences. And so again, there's a responsibility amongst producers to really get a handle on this stuff, and regulators to, kind of guide and ensure people go that way.
It can be allergens, irritants, or even greater
Definitely.
toxicities. So
it's like even
educating the
natural
regulator is
terpenes.
key.
It's like we have some real high natural terpenes, full spectrum products that, you hit it and you sneeze, and that's just all there is to it.
And I think as an industry, it's going to be very important that it's not the regulators or kind of the anti-cannabis people that discover this stuff and jump up and down about it. These type of discoveries need to happen in the industry, and then we need to be proactive in terms of setting the appropriate regulations to make stuff safe, right?
100%.
Right.
First of all, we've been great about listening to all the things I want to talk about, and you guys have done great putting up with me. This is my first panel moderating. I want to make sure, though, that we give you guys everything you'd like. And so we have about 10 minutes left, and I think I'm supposed to land this bird on time. So I think we should maybe open up the floor to anybody have any questions for the panel here that they'd like answered about terpenes or any of the things we talked about tonight. Mr. Frenchy Cannoli.
Actually, Frenchie, it's amazing to have you here. One brief response. I know, of course, Jeffrey spoke to earlier, a molecule is a molecule, and that's one thing that would be said. In terms of one of the most interesting studies I've seen about the effluent of an extract process was authored by Dr. Michelle Sexton at University of Washington last year, And it illustrates that what you're seeing at the end of a supercritical fluid extraction is actually profoundly different than what you're seeing in the whole plant matrix. And so there's a lot of transitions going on. And this kind of brings up, for me personally as a consumer and patient activist, that there's also a huge distinction between pyrolyzing flower, burning it, and vaporizing flower, and a strategic way to flash.
Oh, for sure. We
this
go from
chemistry in a whole
400
plant
compounds
fashion.
to 4,000 compounds when you light it on
I
fire.
personally find a pretty distinguished effect. As nuanced and subtle as it is, I can elicit far more desirable, controllable, beneficial effects with this lower temperature anyways. Michelle Sexton's
So it sounds like the
paper is only one
molecule
I'm
is the
aware of.
molecule, but what comes with it can definitely
Right.
vary, not only from how the product was put together, but how the plant that it came from was grown, and then how the process went down. I
Exactly.
know with CO2 specifically, it's like there's a lot of stuff you do ahead of time with decarboxylating the material, and and then there's processes you'd have to go through to capture those terpenes and then reintroduce them to the product. And out of your 130 terpenes, how many you're actually getting? If people do and have to de-wax their products with alcohol, and then you got to get that alcohol out of there, and heat. Terpenes volatilize at room temperature. This is why we can smell them, and so that could probably have a profound impact on that profile.
Definitely. And if I get limonene from orange juices or orange peels, and I get limonene and only limonene from cannabis, they'll look the same on the chromatogram. So
think
you
one-
can see that exactly same molecule. Yep.
One thing just to point out, though, that can be different between, especially botanically extracted, whether it's cannabis or another plant, is the distribution of isomers of a given terpene. So many terpenes have multiple isomers, and for example, you could buy farnesene that was botanically derived that was a single isomer, or you could buy farnesene that was 6 isomers, and who knows how many isomers you have in the plant that you're extracting. So that is one distinct difference, I think, between limonene, say, and farnesene, and those terpenes.
So it's really
that
how
have
these terpenes were
isomers as
handled
well.
industrially, or
Right.
how they were extracted industrial.
The label will say farnesene
Mr.
mixture
Rosenthal.
of isomers.
That I think, yeah, for sure, the way that we're going to be getting these compounds of interest.
That was
I can't
part of what
wait.
John was talking
That'll be
about, the
fantastic.
dichotomy of, one versus the other. Is it all plant? Is it, farmed out as one plant producing a compound, or cultivated or cultured out of yeast? So I think you're right. Yeast techniques, microalgae techniques, or other types of biotechnology approaches to grow one molecule at a time in a farming of tanks, I would say,
What-
is very different than fields of farms.
One thing I'll say on that, though,
Oh, no.
Correct.
Yeah, it could be many at a time.
one thing I'll say on that, though, that we've run a bunch of economics on that, of course. This plant's one of the most efficient plants in terms of being able to create complex molecules, and we haven't really unlocked the potential associated with the plant and how much it can actually produce. We think that, ultimately, we actually think the plant is capable of producing at least 10x more than what it currently does. And you can do this year-round, and so ultimately, then it becomes a cost equation, right? If we really look at kind of the large commodity-based part of this world and what this looks like, I think that, so there's an argument the plant still could be very competitive going forward, but you need plants that are going to be producing much higher volume than what they're doing today, and probably isolated on a very specific compound if you're going to be able to do that. And that's mostly going to be like genetic editing work that's going to allow the world to get there. And I think then the world has to decide, where do you want your compounds from? Do you want it from a plant that's going to be genetically edited, or do you want it to come from yeast?
Well, and typically, yeah, I mean, as with any other evolution of technology, when we know better, we do better.
One
So
other
great.
thing I'd like to say is that there's a big picture where most of the world doesn't have access to pharmaceutical drugs. This is a plant that, of course, can continually be grown everywhere, and that in reality, we can go so far towards doing so much good for sick, poor, and dying people internationally with a plant that will not have access to a $40,000 a year prescription for a stabilized, consistent,
We're
standardized
definitely
product.
showing that that's economically
And
viable, for
I believe
sure.
that we're coming from a tradition in California especially that is deeply rooted from the original work of women advocates for medical marijuana and so forth, that we can take care of people right now without all of the science proceeding proceeding, we should proceed with all of the science. It's not an either or, but there is a long-term economic implication about where we go that I think really matters.
Most definitely. Thank you for that. And yes, in the very back. Yes, ma'am. Fortunately, there's a video camera right behind you. Well, PubMed is now a great resource. There was a time just a few years back that there was maybe 900 articles on PubMed, and now we're probably adding 10 times that every year. So we're really living in an exciting time.
Probably all of our websites
Yeah.
have some stuff.
Yeah, for sure.
PubMed,
Green
PubMed,
Farm.
Project CBD, the answer page, they have a lot of cannabis-based things.
I tell you
as
what,
well.
come on up after we're all done here, and we'll be happy to point you in the right direction. Yes, sir.
My question is related to the consumption methods that have been touched on. Vaporization feels less harmful to my body, but the reality, I think you might be able to shed some light on, I suppose. I mean, I'm aware that when you bust the plant, you expose yourself to.
I think that there are definitely known health consequences associated with smoking.
I was looking at the little rule in my cologne the other day, going, that's exactly what I,
That's what they're
stuck
cool.
in my face.
Understood. I don't know. Does anyone have any?
Look for studies, I find nothing.
Okay, I have one.
I think that's
One
it's a
basic
huge-
observation is that at the Cultivation Classic, we've had to come up with means of evaluating diverse material from subjective perspectives. Right? We have all these different people with opinions. So what we've chosen as a baseline, in addition to whatever other mode of consumption, is a specific temperature for a terpene kind of aroma profile that actually gets closer and closer towards more objective evaluation. And I tell you, if you try to burn that material, if it's a pipe, a joint, a bong, or any other way, you're absolutely going to be incapable of discerning all of this nuance of flavor. And I would assume that the expression of that flavor is correlated with the fact I can speak about my own personal experience, but the reality is I haven't seen enough science dedicated to the distinctions in the modality of consumption orally.
It's easy if you think about it in terms of THC, right? So if you inhale THC, you get THC directly, but if you orally consume it, you're feeling 11-hydroxy-THC. So route of consumption, method of ingestion is going to impact how you're actually going to feel that molecule, because
100%.
your body might change that molecule to something else. So
Well, hey,
what
everybody.
exactly are you feeling?
Right, oh, and if I may, with you two here, if the
You may quickly.
if people are adding terpenes into the products that are orally consumed, which I'm increasingly see, so we see sativa-specific, edible, et cetera. The reality is the bile. I mean, do these compounds
Endohepatic recirculative cycle.
Yeah, what are the blood levels versus inhalation
Yes.
or anything like that? I don't think there's much of any study on that formally yet.
Well, it seems like there's a lot more to talk about. I mean, I will encourage everybody to continue this discussion outside. I'm not sure if they have someone coming in right after here, but definitely want to respect New West Summit and thank them for putting us all together. I'm going to be here all weekend. If you see me, feel free to tap me on the shoulder. And I definitely hope to see all of you at this rest of this fantastic event. Thank you so much for your time today, and thank you all for coming to be a part of this.
I'm so glad to have made it to your talk. It's fantastic. Yes. Are you going to be here tomorrow? Okay, so just today. Do you have a card with you?